sharing your work. If you're interested in sharing your work in front of a friendly audience, we please contact myself, dbonnell at usgs.gov. And in the meantime, I'll drop here in the chat a link so you can see many of our recorded previous Corrigoning webinars. That takes you to our Great Lakes Cisco's.org webpage, and from there's a link to the YouTube page where you can easily see those webinars. All right, so I think today's webinar is actually a little bit different than what we've done in the past. It's a series of 10-minute or so talks, all on a sort of a common theme. That is Great Lakes beaches as nursery habitat for Lake Whitefish or Atikamekw. And sort of each of our 6 speakers will be sharing not only the results, but also key lessons learned. And then we're really excited at the end of this for to answer questions as a large group. And so that is our plan for today. I want to briefly introduce each of our speakers. I'm just going to introduce them now so as not to interrupt the flow as we go through our 6 talks sort of consecutively. Our first is Kay Skubig. Kay got her Bachelor's of Science in Fisheries and Wildlife Management at Lake Superior State University. She then got her Master's in Biology at Central Michigan University and is currently the Great Lakes fisheries biologist at Little Traverse Bay Bands of Odawa, where she has been working at some capacity since 2018. Following Kay, we'll have Jon Doubek. Jon got his bachelor's in biology in the program of the environment at University of Michigan. Then got his master's at the School of Natural Resources in the Environment at the University of Michigan. Went to Virginia Tech, where he got his PhD in biological sciences and limnology, and is currently the associate professor at Lake Superior State University. Been there since 2019. Following Jon will be Steve Pothoven. Steve got his bachelor's in biology from Augustana. I'm not sure how you say that, Steve. You can correct me if I'm not saying it right. That's a liberal arts college in Illinois. He got his master's in fisheries from the University of Minnesota. And Steve has been at the NOAA Great Lakes Environmental Research Lab, Lake Michigan Field Station for 27 years in some capacity. So I did not know it was that many years, Steve. Very cool. Following Steve will be Cory Brant. Cory got his bachelor's in fisheries and water resources from UW-Stevens Point. He then got his master's and PhD in fisheries and wildlife at Michigan State University. Cory has been a fishery biologist at the USGS Great Lakes Science Center here in Ann Arbor since 2020. Following Cory is Nikki Berry. Nikki got her bachelor's in science and biology at Ohio Northern University, her master's and PhD in biology at Miami University, and she has been a fishery biologist at the USGS Great Lakes Science Center, now stationed in Cortland, New York, since 2024, since last year. And then finally, following Nikki is Jim McKenna. Jim got his bachelor's in bachelor's in marine science at Stockton State College in New Jersey. He then got his master's and PhD in biological oceanography from the University of Rhode Island. And Jim has been a research ecologist at the USGS Great Lakes Science Center, also in Cortland, New York, for 30 years. Congratulations on that milestone to Jim. So really excited about this group that we have. I'm going to be navigating the slides. And so any mistakes are my own in terms of pace, and we will see how this goes. Kay, you are up first. And I'm going to close my audio.
All right. Thank you for that introduction, Bo. Kay Ndishnakaz, Hali Ndonjaba, Pataskin Ndora, Ndonjanoki, Dikamig, Minwa Atonapi. That's my introduction in Anishinaabe Uh, to start off these talks today, we're going to be getting kind of a background on what led to all of this. Other talks, following me will be about specific projects and analyses studying Dickameg in these, larval habitats. And I'm here to set the stage for those talks and share a history of how we all got here and to show some data to explain why this work is so important. Next. There you go. So Dickameg Lake Whitefish are incredibly culturally important to the Anishinaabe people, including the Little Traverse Bay bands of Odawa Indians. There's a continuous, unbroken, and ongoing relationship between the people and the fish, this relative. I'm sure others today will share the economic value of this species, but I'm focusing on the relationship and the importance of ensuring that it continues for the next 7 generations and beyond. Unfortunately, this relationship is in trouble. Dickameg have faced population declines from a myriad of factors, including non-native species and habitat destruction and degradation. They're also facing potential bottlenecks at the larval stage. So knowing that these fish are so important and that they're facing these problems, what do we do about it? Well, the first step to solving any problem will be to first understand the problem. Next. That first looked like uncoordinated beach sampling, where agencies throughout the Great Lakes region were doing their own sampling completely independent of one another. And LTBB was one of these agencies. We started Newsta netting in 2012 along the beaches in this area. But that uncoordinated beach sampling, it wasn't quite enough. And our previous program director, Kevin Donner, recognized this, and that was the start to something more. Next. These fish need us, and in turn, we need one another. In the beginning, the intention of a coordinated beach sampling program was to determine if we can create any kind of meaningful age zero year class strength index. So in 2013, that's what was done. It was the first year of coordinated beach sampling for Dickameg in the Great Lakes. And this was done by Little Traverse, Bay Bands, Charlevoix, DNR, and Sioux Tribe. So these 3 agencies had a total of 19 sites. And from this map here, we can see that most of these sites were fairly close together and in northern Michigan. The coordinated beach sampling had a pretty slow start, and participation was and still is completely voluntary. It took about 2 to 3 years before this concept really started to gain traction and extend, further beyond those original 3. Kevin Donner, the previous program director here at LTBB, was one of the folks that headed up the original push for that coordination, standardization, and expansion. There are so many individuals that made this coordinated beach sampling possible, but I'd like to give some special acknowledgments to Mark Ebner at Sioux Tribe, Nathan Barton at Grand Traverse Bay Band, Jason Smith, who is at Bay Mills now, but has been at LTBB and Sioux Tribe, Dave Carafino with the Charlevoix DNR, Ted Tresca with the Great Lakes Fish Commission, Bo Bonnell with USGS, and Cory Brant, also with USGS. Through all of these efforts, the coordination from beach sampling have gone from these 3 agencies and 17 sites to this. Bo, next. There are 9 agencies and counting, and beach sampling sites all throughout the lower Great Lakes. I'd like to again acknowledge Cory Brant for making these maps and also for heading up the newest push in coordination efforts. I wasn't around when all of this coordinated beach sampling was kicking off, and I did my best to retell the story as it was told to me. If I've missed any pieces or people in this background, please let me know. My email will be on the final slide. I'm very thankful to Bo for putting this webinar together, getting us all here, and for asking for this background to be shared. Next. So now that we know where we all stand, and that's together, I'm looking to highlight some of LTBB's work. So we've been doing beach sampling since 2012, 2013. That involves Newston as soon as the ice is out. Although if you look at this middle picture here, we can see Chad Lefevre standing next to a sheet of ice. The ice doesn't have to be completely gone for us to start our field sampling. And it continues through the summer with seining until those fish are big enough that they've left the beach. Next. So we have four sites highlighted on the orange box over on the right of Cory's lovely map. So these four blue dots, starting from kind of that bottom left and working its way up and around the tip of the Lower Peninsula, we have Little Traverse Bay, Sturgeon Bay, Big Stone Bay, and Duncan Bay. And something very interesting that we're going to track as we move through this is that the Dickameg are known to have boom and bust year classes. Next. So we start by looking with our Newston catch per unit effort. So on our Y, we've got catch per unit effort in meters cubed, and on our X is year. And what stands out to me immediately are two huge spikes that we see relative to the rest of them in 2013 for Big Stone Bay and in 2014 for Sturgeon Bay. Now, with these being so much higher than the rest, it does make the other bars a bit harder to see and to really understand maybe what's happening in those other sites in those other years. So let's change that to visualize that better. So in this one, this is the same graph as before, just with our y-axis cut off at 4 instead of 16. The two bars with the asterisks above them indicate the ones that would go up to 16 plus if our graph extends that far. Now, let's focus now on these other bars, the smaller ones that we wanted to get a better idea of what's happening here. We can see almost rhythmic peaks and valleys as we move through time at these different sites. And this is what we would expect in boom and bust life cycles. But we also notice that our highest highs are getting lower and our lowest lows are lower still. So yes, there's boom and bust, but there's still trending downwards for our catch per unit effort. It's declining. Next. Out of curiosity, we can look at average length and see what's going on with the fish at these sites. And here we see generally what we expect to see. We sample it ice off and continue until the fish are large enough that we have to switch to a seine. So generally it's about between 15 millimeters and 23 millimeters, and our average is all within there. But we do see some interesting year-to-year shifts in the average size, which could be run down in the future to determine if that's a sampling artifact, if we were sampling later, water temperature is warmer, things like that, or if length is truly changing. It could be because temperature is warmer. A lot of different factors affect that. Anecdotally, I do believe that we see flips in not only which site has the highest catch rates, but also variations in size. Next. Now, when the fish grow large enough to outrun the Newston, I told you before we switch to seining. So here in our seine chart, we've got our catch per unit effort in meters squared on the Y and our year again on the X. What's really interesting to notice here is that we don't see the same spike in 2013 and 2014 as we did in our Newston net. And we see a comparatively large spike in 2020 that we don't see on our Newston. Despite those things not matching up, I believe that this seine chart does do a better job of showing these declines. There is some boom and bust, especially with that one really high 2020. But overall, if you look at everything other than that, we are seeing lower catch per unit efforts across the board, especially looking at that 2013, 2014, 2015 to then 22, 23, and 24. Next. So let's look at length again. Pretty similar trends to what we saw with our new ston. We expect that these fish become vulnerable to the seine a bit over 20 millimeters and are gone from the beaches by about 50 or 60 millimeters. I think it's interesting that we see no one bay having consistently higher average size, and instead we see variation. Just like our new ston, this variation is something to parse out. Next. So what can we learn from all of this? Well, we've learned that seine and new ston, they don't always match. We also know that catch per unit effort seems to be declining despite boom and bust cycles. There have been more busts than booms, and the booms have not been true booms. We've also seen that of our four sites, Sturgeon Bay and Big Stone Bay have the highest catch per unit effort, although high is still very relative. The variability in catches really highlights the importance of having a long-term data set that's sampled year to year. Let's imagine that we had only sampled 2013, 2014, and 2021. Had we only done those 3 years, we might believe that these fish are doing amazing, but they're not. And had we only sampled Duncan Bay in 2022 and 2023, we might have believed that there are no whitefish using that area as nursery habitat. But we got a signal in 2024. We found whitefish. It wasn't very many, but they were there. And so the variability that we see in these fish truly makes long-term data sets really important. Now, in the beginning of this, I said that the original intention of this coordinated study was to determine if we can create a meaningful age zero year class strength index. This is a very tricky goal to meet. But the seemingly simple beach sampling work does set the stage for other analyses done by individual agencies as well as the collective. Paired day-night sampling, UV exposure, identifying nursery habitat, it all gives us a better understanding of this species and it better equips us to solve the problem and ensure the relationship with Dick and Meg continues. Next. You can go ahead next again. So I'd like to leave this with, just all of these things highlight truly the importance of long-term data sets and continued partnerships. This is good and important work that needs to continue. Like all science and every other part of life, we're all stronger together. Thank you.
Okay, I'm going to break up what I said I wouldn't do. I just want to quickly jump in and say thank you. You kicked that off beautifully, and you've set the stage really nicely for this next set of talks. So, job well done. All right, on to our next talk. Jon.
Thank you, Bo, and thank you all for tuning in for these talks here today. I'm a faculty at Lake Superior State University, and I'm excited to be here to give a lightning talk on behalf of this project and the team and individuals you see here on this first screen. And this was a project ongoing for the last few years to look at factors affecting larval lake whitefish and cragganine abundance in Lakes Michigan, Huron, and Superior. Next, please. So first, just a quick acknowledgement again to all of the individuals and teams on this project, funded through the Great Lakes Fishery Trust and other agencies to make this work possible. Next. So, and I'm just realizing, Bo, as well, I left my animations on here, so you can feel free if you want just to animate to the end of each slide as well. Sorry about that. But so yeah, lake whitefish have experienced population declines in various locations in the upper Great Lakes, particularly Lakes Michigan and Huron. And part of the attribution to this has been the invasion of dreissennid mussels, zebra and quagga mussels, which eat phytoplankton in the base of the food web, and so less food for zooplankton, less food for fish. And particularly a vulnerable stage could be this larval fish, not just like whitefish, but other fishes that need zooplankton as a food source during the first few weeks to months of their life. And so this project really looks at that larval stage. Next. And a big part of this is using a standardized sampling approach sort of as Kay brought up on the last talk, the importance of sort of a standardized sampling, kind of large scale comparisons to compare these things across different regions. So lake whitefish larvae, when they hatch, they're commonly in nearshore littoral habitats, and they could be right on the beach. So in 1 meter depth or less of water defined here, or in the near shore, maybe 1 to 15 meters or so depth. So in these near shore regions. So agencies have been sampling at various depth locations depending on protocols. And typically during the daytime, fewer humans are nocturnal. So generally, logistic wise, of course, we sample during the day. But of course, their prey zooplankton as well as larval fish can exhibit diel vertical and horizontal migration, and estimates can be higher at nighttime. And so part of this project is really to look at any differences day and night and how that could affect our interpretations of their abundances. Next. So a part of this project in 2023 and 2024 was a field research component where teams sampled sites on the beach and some at day and night, and also a subset of those on the near shore. And we're looking at zooplankton density and biomass and larval lake whitefish density and day and night comparisons between those. And there's other questions associated with this project, but for just the time purposes here, this will be the main focus. Next. The other part of this I wanted to highlight is a mesocosm research component as well, which the first year of this was in 2024, and we're about to do this again in 2025. Where at Lake Superior State Center for Freshwater Research and Education, there's a mesocosm facility set up with different tanks and getting water and zooplankton from the St. Mary's River at a time where the zooplankton is similar to what's, the lake whitefish when they hatch. And we're feeding them different treatments. So inoculating some tanks with low zooplankton density, with medium zooplankton density, and higher zooplankton density, using densities commonly observed sort of at these comparisons in the field during this time and location. So looking at how density or the food items would affect their growth, condition, and survival. Next. So to jump into some methods, and part of this is to sort of set up the project and the objectives and what's been done, and then give some preliminary results after that as well. So agencies hit the beach right after ice off to sample for larval lake whitefish. And across the 12 agencies on this project had a pretty good spatial coverage in Lake Superior, Michigan, and Huron. Sampled collectively over 50 sites, up to 3 times each during the day, which was our baseline. So about ice off to, or when it starts to warm up, to up to 6 weeks after that. And then a subset of those were sampled at nighttime. We had over 30 of those sites also sampled at nighttime for each year, up to 3 times as well. And those were paired, so within 24 hours of each other. And then from those beach sites, approximately 15 of those were also sampled day and night on the near shore. And these depths range from about 1 to 10 or 15 meters depth. So there's a different sort of gradient of depths for those as well for comparison. And again, these were in 2023 and 2024 for the sampling. Next. So for the standardized sampling protocol, across all the different areas, we sampled the same way for, substrate environmental characteristics, water quality, chlorophyll A for primary productivity. We sampled zooplankton with horizontal zooplankton tow nets shown there, and then also sampled for fish with Newston nets. If you could hit next, please, to show the one back. Sorry, a little back. Thank you, Bo. Sorry for the animations. With the Newston net shown there, pulled by 2 people for 20 meters on the beach for larval fish. And then for the boat sampling, it was similar, but via boat, there are a couple differences, such as a longer tow for the larval fish, but also using a Newston net, so using similar gear both on the beach and in the near shore. And all of the fish were counted, and up to 25 were collected to take back to the lab for each sample. Next. And then back in the lab, zooplankton were counted and processed. Larval lake whitefish were identified to order or carrageenine versus non-carrageenine, and separated total lengths, and all of that was measured. And not a part of this talk, but I just wanted to highlight, we're also extracting the stomachs to see what the diet composition is and how that might relate to what's in the water column. And so far about 1,500 fish have had diets analyzed so far for this project. with many more fish yet to go. But the other part of those fish that have the diets extracted, the other part is sent to the U.S. Geological Survey in Ann Arbor for genetic classification down to species, which is super important to know if it's a lake whitefish or a Cisco, for example. So a big shout out to Amanda Ackes, Ann Ropp, Alicia Alvarez, and others on the team there for all of the help with genetics, not just for this project, but all the other projects out there. And also just also a special shout out to these individuals: Ethan DePass, Silas Dunn, Simon Freeman, Ellie Pro, Gwen Phillips, and Mason Spees. These are former or current undergraduate students at Lake Superior State University who have not just spearheaded the field work, but also the lab work in processing the fish and the zooplankton to make this all possible. Next. So first looking at density to get in some of the results here. Again, preliminary analyses so far. About 100 zooplankton samples have been processed so far. And this is density on the y-axis at day and at nighttime. And most of these are on the beach right now, so in the really, really shallow waters. And then also biomass at day and night. And nighttime density is about 2 times higher than day, and biomass is about 3 times higher at night versus day. But as you can, if you're think about zooplankton a lot like I do, the y-axis, that's pretty low density. Oftentimes there's fewer than 1 zooplankton per liter in these waters. And most of the specimens were copepods. Next. And then for a cruganing count, this is just on the beach. Again, really quick first glance at this. And the y-axis is how many cruganings were found in a 20-meter tow Houston net. So that's the number on that y-axis. And so far at nighttime, we're finding, again, this is a really just coarse assessment. We want to get into some more paired estimates by site. But, on average, nighttime is about twice as high than daytime for cruganing count. Next. And also comparing the catch per unit effort of cruganings on the beach samples versus the boat samples. And now this is standardized per meters cubed of water because the boat towed more water volume through than the beach, so standardizing for the effort here. But we found so far about 25 times higher catch per unit effort of cruganings on the beach versus boat samples. And this is irrespective of day versus night, so those are together. Next. And then really quick on just preliminary genetics data. This is for about 600 individual fish so far, with many more coming up. And just very, very roughly grouped by region. The yellow is dominated more by Lake Whitefish, and then the purple or pink is dominated more by Cisco. So many locations, and there's a lot of gaps. We're getting data, sending data for the data are being processed here soon, but many places so far dominated by Lake Whitefish, with Grand Traverse Bay dominated by Cisco. Next. And so for the mesocosm experiments, real quick, just to sort of orient you, the gametes were collected from Lake Michigan by Little Traverse Bay bands, and then we received those up at Lake State. And the photo there is of the Center for Freshwater Research and Education's mesocosm facility at Lake State. So you can kind of see the tank setup and the size. And the water's from the St. Mary's River, and we get zooplankton. We basically have a zooplankton farm on the St. Mary's River, where we just get zooplankton and put them into tanks to mimic low, medium, and high zooplankton treatments, and then add fish and run the experiment. So last year we had four replicates of each treatment for a total of 12 tanks, and we're about to start the process again here soon in just like a few weeks. Next. So results from last year for the feeding experiment, the low being 1 zooplankton, medium being 5 zooplankton per liter, and the high being 20 zooplankton per liter. We found that more lake whitefish survived, still not a high percentage, but more lake whitefish survived in the high food treatment versus the low and the medium. And not shown here, but the average length at the end and average weight at the end were also trending higher for the high treatment as well. Next. So yeah, just to wrap up, a few takeaways. Just I think the importance of standardized sampling at a large scale to really, quantitatively compare estimates across the different locations that one samples for abundances. Thinking about day versus night differences, are there differences? If so, how much, and what does that mean for our understanding of their abundances? And, There's places we know that have been hotspots, historically for larval abundances and for potential spawning, but identifying those and also potentially new areas that have not been sampled before are also important. And ultimately, a big goal of this project is to look at zooplankton density and other environmental factors and the relative importance of these for larval lake whitefish to survive into juveniles and beyond. Yeah, thank you all for tuning in, and I'm happy to take some questions in a little bit.
All right, thank you. I'm Steve Pothoven, and I'm going to be talking about some of the NOAA sampling that's taken place in Southeast Lake Michigan. And these are some of the sites that Kay showed. We've been doing this since 2014, and we've been doing both Newstone nets. We have a couple sites located in and outside of the harbor at Muskegon. And then we have 3 of our beach seine sites that are located off the river mouths in Lake Michigan here. This work was mostly done to look at the feeding ecology and be as well as be part of the larger sampling effort. But today I'll be talking about some of the actual results from the catches. And as far as we know, all these fish are lake whitefish. We haven't had any reason to believe that they aren't all lake whitefish, although there are Cisco moving into the area, so that may change in the future. Next. So just looking at some of the catch data, just number per haul for both the Newstone and the beach seine, and you can see when there are fish there, it's quite variable in those years. And so the variation here is among days that we sampled. We sample weekly with the Newstone and over 2 or 3 weeks for the beach seine. So you can see that the years that we had fish, there's still quite a bit of variability, although it does look like some of the years, especially 2015, was a relatively good year. And then most of the last few years have been really poor. They're not variable just because we haven't caught hardly any fish in the nets. Next. Now, one of the reasons to look at the early life stages of fish is to see if that can give us an idea of how many fish there will be in the population in the future. And so one of the things that we've just started doing, tackling this is the Minnesota or Michigan DNR has some sampling that catches fish once they're age 3 in this region. And so we're looking to see how the early life stages compare to what they're seeing once those fish are age 3. And you can see it probably covers those first 3 years of our early life sampling. And those fish, which were our good years in our Newstone and beach seine, really don't appear to show up once they're age 3 in the DNR trawl surveys. And so there's a couple things that I can think of. There's probably more that could be happening. One is between the time that those fish are caught on the beaches in the Newstone or the beach seine, they're disappearing before they're age 3. The other possibility that could be is even though we have relatively good years in our early life sampling, they're really not that good. And in order for us to see fish once they're age 3, we would have to be catching much higher numbers of fish during the early life stages. So unfortunately, we don't have sampling on the beach in the early 2000s to see if that might be the case. Next. So one of the things that we wanted to look at was what sizes of fish are we catching in each of these gears? And so this is all the fish overall, 9 or 10 years of sampling. And one interesting thing in our Newstone sampling was we really aren't catching those fish until they're 15, 16 millimeters long, once they've lost their yolk sac. Of course, smaller fish should be vulnerable to these nets. They're a 500 micron mesh net, but they aren't showing up in the net, obviously, relative to how many there must be. And so maybe those smaller fish aren't on the beach where we're sampling and they're further offshore, or these are mostly off drawn river mouths or river mouths, and maybe they haven't worked their way from where they hatch onto the beach yet. For the beach seine, we start catching, they seem recruited to this gear at our site anyway, around 25, 26 millimeters. And then by the time they're in the early 30 millimeter stage, their catches are really dropping out. And this is going to correspond to the time when those whitefish, their mouth is changing. they're becoming more benthic and they're moving offshore. So it's really kind of a small size window where we're catching the fish in the beach seine. And if you think about it, we're really probably only sampling one life stage. So between the yolk sac and when they become more benthic, that life stage is what we're sampling. We're getting the early end of it with the Newston and the back end of it with the beach seine. Next. Next slide. Yep. So unlike what Kay showed, we do see relatively good correspondence between our Newston catches and our seine catches, at least relatively for this type of data. We'll probably take that. And like I showed with the size distribution, if we're really only sampling one size class or one life stage of the fish between the yolk sac and when they become more benthic, maybe that's not surprising. Other areas I know catch larger fish or smaller fish, and so that may be a reason where some of these studies don't have as much good correspondence between the seine and the Newston. And next. And then one last thing, we all know that temperature is an important variable for growth, along with food with fish, but a lot of times we don't have good temperature data. If we take temperature where we're out sampling, that's quite variable. From time to time, it can go down or up between sampling efforts. But a lot of these sampling sites are located in areas where there are weather stations, and you can pull air temperature data off from these sites. And you can develop, and because they're so shallow, obviously it seems reasonable that air temperature would really be related to water temperature as well. And so here we see the cumulative air temperature we see a strong relationship between the cumulative air temperature and the length for both the Newston and the beach seine. And so this is one possible tool that we can use to control for temperature or to provide environmentally meaningful sampling. In past studies, you could also use it if you wanted to keep track of the cumulative air temperature and target specific sizes if you know there's specific sizes or more vulnerable sizes that you're trying to sample as well. And that can be important, like for our sampling, we have a fairly small window for the beach seine. And on average, it's probably about 16 days where we could effectively sample those fish. And so you think about our sampling methods of sampling twice with a week in between, we could be in and out of an effective sampling period. So this is one possible tool that can maybe help focus that sampling. And that is it. Thank you.
Thank you. Yeah, I'm super excited to be part of these talks. My name is Cory Brant. I do have to apologize. I'm rocking a solid cold right now, but that's not slowing me down on my excitement to have conversations with this group about beach sampling today. So I'm excited to speak, to give this talk on behalf of a very large team. I'm just representing the work here. And this work was, ties into our Lake Huron CSMI, or Cooperative Science and Monitoring Initiative work, the CSMI work from 2022. Next. I'd like to acknowledge a lot of folks. And here I also want to just point out that I especially want to acknowledge our student contractors, our term scientists, and our seasonal employees that are absolutely foundational to all this work that we do. I also want to thank the funding agencies, Great Lakes Restoration Initiative, and the Correggian Restoration Framework for funding this work, and many others that help us with our sampling designs. Next. So the focus of this work, this was inspired by the foundational stuff that Scoob mentioned at the beginning. We essentially wanted to help monitor larval fish on Lake Huron, spread out the effort with the other agencies that were already doing work. We wanted to add sites, add resolution spatially. But we also specifically wanted to monitor northern Lake Huron versus southern Lake Huron for larval fish on the beaches. And this is due to that large contrasting productivity gradient between those two regions. So the North Channel generally is less nutrient rich. There's less establishment of dry-scented mussels up there. It's a little bit different shoreline habitat. It just tends to be very different. In very southern Lake Huron, it tends to be, I think I said, so the North Channel is more nutrient rich. My apologies. And then the southern parts of Lake Huron tend to be very nutrient poor, very crystal clear water, and more establishment of dry-scented mussels down there. So it seemed like a really interesting way to maybe tease out some of these factors that may influence recruitment constraints for these young lake whitefish in their first year of life. So we had a couple of research questions. I'll revisit these at the end. One, we asked, is there just more diversity in larval corriganines in the North Channel versus southern Lake Huron? There's, adult cisco and whitefish and other corriganine species are known to be up in the North Channel. So we asked that. And then we were wondering, is there greater corriganine densities in the North Channel? Could this be tied into nutrients versus southern Lake Huron? Another question, is there greater zooplankton densities up there to sort of complement the higher densities of fish? Additionally, is there better growth in the North Channel? Is there any signs of stunting in the south where the productivity gradient is, where there's those lower trophic levels are much, much harder to detect? And then also, with less food, if there is less food available, does that mean that there's less food in their stomachs? And would that then help us understand if there's slower growth? So these are some of the core research questions. Thank you. So our sites, just a quick look at our sites. We had 4 sites in the North Channel: Lakeside Beach, Peace Park, Fourth Sand, and Baharial. And then 4 sites in southern Lake Huron indicated there. Next, please. And our sampling approach, as mentioned in previous talks, is very similar. We synergized this with everyone else sampling. So we had a little bit of a difference in that we hand-towed a Newston net on the beach at the same time, at some of those sites, at the same time that we did small boat bongo sampling for larval fish, just to compare the very beach habitat to just offshore of the beach. We took those larval fish. We also did tows for zooplankton alongside our larval fish samples. With those specimens, we did identification and counts. And then with the fish, we confirmed species with genetics, did some morphometrics, aging, and diets with those fish. Next. So overall density is on the beach with lake whitefish. We were a little surprised by this one when we processed all the data from 2022. We actually saw a peak, or one of the highest density sites was in the south, unlike our thoughts that it would probably be in the north. But overall, you see there was real high variation in density at these sites. Beaches are not the same, at least within 1 year. And according to our sampling, we just saw this high degree of variability with our sampling. And Harbor Beach surprised us with these very high densities overall of lake whitefish larvae. Next, please. So this is that same mean density plot that I just showed, lake whitefish per meter cubed across the sites. And I just overlaid it on top of our mean density of all crustacean zooplankton, so number or count per liter. And I just wanted to quick point out with the two green rectangles there highlighting it is that high zooplankton densities didn't necessarily mean high fish densities overall. And it was sort of like one site surprised us at having high densities, very, very low, like Harbor Beach, high densities of fish, but very, very low, orders of magnitude lower zooplankton densities compared to some of the North Channels. And perhaps this doesn't surprise folks, but I thought it was pretty notable to include. Next, please. This is a lot, and it's not for detailed reading, but this is our overall zooplankton densities by groups of certain zooplankton. And I just wanted to point out that I indicated where the 1 individual per liter mark is. And generally in the North Channel, most groups of zooplankton, nauplii, cyclopoids, cladocerans, they were sometimes orders of magnitude higher in the North Channel overall, across time. versus southern Lake Huron sites, with the exception maybe of calanoids, which were kind of just rare everywhere. But I just wanted to point out that higher zooplankton density in the North Channel overall. Next. So I chose two sites to talk about today. I'm just pointing them out there. I'll quickly tell you why I chose those two. It's Fourth Sand Beach in the north and Harbor Beach in the south. Next. Due to time constraints and resources, we had to prioritize a couple of sites to go into more details with these fish. We chose Fourth Sand and Harbor Beach. One, because these graphs just show mean density of fish we caught over time on that beach with the new spawn. These are larval lake whitefish. And you'll see for Fourth Sand Beach, there's this peak around 11 May that gets to about 1 fish per cubic meter. And then they taper off again until we couldn't catch them anymore. Just below Harbor Beach, we also saw similar densities that gradually picked up until that might be a little cut off for you, but I think that's around the 2nd of May. You'll see that higher peak. Oh, there it is. 2nd of May, around 1 fish or a little bit over. So similar densities at their peak. But when we looked at the growth over time, sorry, not the growth, just the general size of the fish over time, we saw in the North Channel, they just continuously got bigger until they disappeared. They hit about the 20 millimeter mark, and then we weren't catching them in the new spawn anymore. While at Harbor Beach, they stayed fairly, consistent in their size as we caught them each week. And there could be a million ways to explain, they could be new batches of fish moving through. It's not the same population down south. There could be a lot of things to think about here, but it certainly sparked our interest to investigate more. Next slide, please. So now I'm going to show a series of graphs in a kind of slide-by-slide animated way that just show the total length of the fish we caught and the estimated age using otoliths that we did in the lab with these fish. I broke it down between yolk sac fish, post-yolk larvae, and then I combined the two in the third graph. So you'll kind of get used to how this turns, the graph is shown here. But generally, and then I'll show Fourth Sand first and then add Harbor Beach as we go. So Fourth Sand, you see, it's a little noisy, but generally as they got older, they were getting longer. Next. Harbor Beach kind of overlap and they do sort of indicate a trend that maybe they're falling below the curve a little bit in Harbor Beach, but it's pretty much overlapping. Harbor Beach yolk sac larvae generally, getting longer as they get older. Next. In the post-yolk larvae, they have absorbed their yolk sac at this stage. A little noisy, you can see, but generally the North Channel fish were getting larger and aging still. Next, please. And here's where the Harbor Beach fish tend to really start falling below the curve in the post-yolk fish, where they're still, getting longer as they're aging, but they're just below the North Channel fish. Next. So when we combine those two, next again, just turn them both on here. You'll see that there's some indication that the Harbor Beach fish are falling below the curve. They may be stunted down there. Something is sort of slowing them down compared to the North Channel larval lake whitefish. Next. Then we looked at the maximum diameter of the otoliths, just another proxy that we could use for age. That work was inspired by some work from Aaron Dunlop and Isaac Herbert, and some other papers by Oyadumari and Auer in 2007, and some other colleagues too inspired us to take these measurements. So you can see, in the north, generally they're getting longer as that otolith gets larger. Next, please. Harbor Beach fish, even in the yolk sac stage, seem to be falling below the curve here, where their otoliths are getting bigger. They're getting longer, but they're just below the curve of the North Channel fish in the yolk sac stage. Next, please. Here we have post-yolk, same graph. Otolith getting bigger, length getting bigger. Next. And again, in the post-yolk stage, those Harbor Beach, Southern Lake Huron fish are below the curve. Next. And next. So you can see there that it also is just another indicator that these fish might be, they certainly seem stunted in Harbor Beach compared to the North Channel. Next. Another thing we looked at is just another way to look at that data is overall the otolith max diameter as a proportion of total length. You'll see right away in the yolk sac stage that harbor beach fish have that higher proportion. That higher proportion in harbor beach fish means their body was shorter in proportion to their otoliths in this case. In this case. And the significance when you combine all the larvae is actually driven by those yolk sac fish. So this makes us wonder, are they starting out a little smaller? Is something wrong while they're hatching? Just generating more research questions for us to dig into here. Next. Here's a picture of a larval otolith. I won't go into too much detail, but hopefully we can discuss some of this. This one happened to break, but it has really good viewing windows here. But one thing I just want to point out is that you'll see some dusky bands kind of barely showing up towards the center. According to the literature and colleagues, these definitely, they usually indicate the hatch point, those dusky, thicker bands. And then a little ways out, as you move out from the center, you'll see two more. Those generally are considered yolk sac absorption check marks. And check marks are essentially like a slowdown in growth. Could be due to something stressful, a cold water shock, a hatching event, absorbing yolk sac, that kind of thing. But in this yolk sac, in this otolith, you'll see several more check marks. And so we actually counted these up for each fish. Next, please. And when I summarize the number of check marks per otolith, which could indicate the number of stressful events these fish go through, in the yolk sac fish especially, they seem to be more variable in their check marks compared to those North Channel site fish. And again, the significance across all the larvae is sort of rooted in these yolk sac fish. So this is kind of exciting to see and is also generating more questions for us. Next. So to check in on the questions, more Corriganine diversity. Yes, we saw that. We got Cisco larvae and round whitefish and Lake Whitefish and a lot up there. Down south, we just caught Lake Whitefish. Question 2, greater densities, not necessarily in the north versus the south. We saw the highest in the south, so that surprised us. Question 3, greater zooplankton in the north versus the south. Densities, yes. Question 4, that better growth in the North Channel versus Southern Lake Huron, it appears so. And then question 5, less food available means less in the stomach and slower growth. We are working on this one right now, right in the middle of our diets. And so that will be forthcoming. Thanks so much for the time, and I'm excited to hang on and have discussion with the group.
Hi, everyone. Hopefully you can hear me. My name is Nikki Berry, as Bo had suggested, and I'm going to talk to you today about some of the implications of larval lake whitefish exposed to UV radiation in beach habitats. I want to take a moment, thank all of our funding sources, as well as my many co-authors that are listed on this project and really helped with the data collection and analysis, and Miami University, where I was stationed for a while before coming to the USGS and continuing this work. Next slide. So today we're going to talk about UV radiation, or ultraviolet radiation, which is the damaging wavelengths of sunlight. This is the stuff that gives us people sunburn, especially varying amounts depending on how much protection you may have, whether it be sunscreen or pigment in your skin. It also can cause sunburn on fish, and many of you may have seen this slide to the right, where we've recently, with the help of Jen Schumacher, who's at Miami University, we've recently developed ways in which we can actually quantify the level of sunburn on larval fish after being exposed to UV. Next slide. From my dissertation and earlier work, which many of you may have seen a slide like this before, we found that the UV tolerances, or the lethal exposure level at which 50% mortality would be expected, can vary by the species of Corriganines. For example, larval bloater have almost 2 times or more than 2 times as high of a UV tolerance than larval lake whitefish, and Cisco are somewhere in the middle. And this damage can have longer effects. It's not just lethal, but the picture to the left here, these are fish that are actually technically still alive, as in they have a heartbeat, but you can tell from the morphological damage onto these fish. In reality, these fish would not survive in nature. Next slide. We did the laboratory experiments establishing what the differences in the tolerances were, and then we started to expand this and extend this research to looking at what solar radiation and the interactions with these fish. So the first experiment we did was really trying to test this thought that the larval cragganines are positively phototactic, and that just means they're attracted to higher intensities and visible light. And if that's the case, they may be susceptible to solar ambush from the sun, which was a hypothesis originally proposed by Craig Williamson in his 1995 paper. And it's effectively that fish that are attracted to higher intensities of light or the sun may occupy shallower depths within the water column, get that bright sunlight for whatever reason they may be attracted to that. And if they cannot detect the presence of UV radiation, especially in a really clear habitat, they would be ultimately swimming to their deaths. So we did these binary choice experiments. We did 2 different kinds. The first, we removed UV radiation altogether, and we only manipulated the intensity of light. So the yellow circle on the left-hand side of that first kind of conceptual diagram, that's going to illustrate our high intensity of sunlight, and then our low intensity is by the dark gray. The second set of experiments that we did, we wanted to test if cragganines, so Cisco and Lake Whitefish, could avoid UV. So we kept the light intensity of visible light equal on both sides, but on our left, not always on the left, but in this diagram on the left side, the purple is represented by the side that had UV present, and the dark gray is where UV was absent. Next slide. In doing these experiments, we tested 2 populations of Cisco and 2 populations of Lake Whitefish. Our 2 populations of Cisco came from one from Lake Coronis, which is an inland lake in Minnesota, and the other from Lake Ontario. It's actually a population that we caught from Chameau Bay. And what we found was that for Cisco, which I have out kind of boxed in green here, they tended to seem to prefer slightly lower intensities, if not no. Preference was really made for our Lake Ontario population. And opposite of that, with our Lake Whitefish experiments, we tested a population from Lake Michigan and a population from Lake Superior. We found that Lake Whitefish tended to spend more time on the higher light intensity sides, just in maybe a positive phototaxis response. Next slide. Interestingly enough, when we removed the differences in the intensities and we only manipulated the presence of the wavelengths of light, specific wavelengths, we found that Cisco overwhelmingly spent more time on the UV absent side of the trays, and that was true for both populations that we tested, whereas Lake Whitefish made relatively no choice, if anything, maybe slightly more time on the UV absent side, but for the most part, there was really no choice made in either of those populations. Next slide. So really what this is driving home is just building that background context here, that Lake Whitefish in particular, they have a low UV tolerance, and they might be attracted to these higher intensities of light. As many of these other talks have said, even Jonathan's, were catching higher densities or more cragganines potentially at night. So this is a little counterintuitive, but with the lower UV tolerance and the inability to differentiate between UV wavelengths, the presence of it, maybe this is causing them to be more susceptible from solar ambush from the sun. In other words, getting sunburned and potentially dying. Next slide. We wanted to connect these laboratory experiments that we did to some more field surveys in shallow beach habitats. And this is a more recent element of the project. So I just have some preliminary and kind of a small sample size of data to share with us today. Next slide. So to summarize, these are all the sampling events that we've done, and actually many of the sites that were talked about today are included in some of the sites that are presented in this figure. Generally speaking, these beach habitats are very shallow. If you think of them as being a meter or 1 to 2 meters, generally a meter or less deep. And of all of our sites that we had sampled, 69% of them were actually had a UV depth, which is we measure it by a 1% UV depth. You can think of this similar to a 1% PAR depth. And we estimated it to actually reach deeper than what the actual bottom depth of that site is. And so that essentially just means that UV is penetrating throughout the entire water column. So this is possible for a handful of the sites that we had sampled. Next slide. Looking at just a subset of sites as one example here, and trying to pair it with some of the larval fish. Cory already had talked about this, but his three of his southern Lake Huron sites, we also took UV measurements at those beaches. And what we found on the left here, again, low densities of Lake Whitefish were caught throughout the whole sampling season. But with those changes in density, so that decreasing density going from Harbor Beach to Port Sanilac to Port Huron, we see an inverse, in some extent, relationship with an increasing estimated 1% UV depth. And so there's potential here. If nothing else, this just does show some support in which UV may be one of the players. Next slide. And so I just kind of want to end it on that note here. And just thinking back to what Kay so elegantly put earlier to start this talk is, we're obviously not saying that UV is the end all be all factor, but we definitely are finding some evidence and support here that for Lake Whitefish in particular, UV may be one of the contributing abiotic stressors for these early life stages of fish. And with that, I'll pass it on to Jim.
Hello, folks. Appreciate the opportunity to present today and to have the last word, so to speak. Also want to thank the previous speakers for setting things up so well. There's a number of themes that are going to come through that I'll re-highlight as we go. But there's a lot to cover, so I'm going to dive right in and start by saying something that we all know, and that is that Lake Ontario gorgonids have been extirpated or severely depleted. And Chameau Bay and the Bay of Quinte in the eastern end of Lake Ontario are our last remaining significant spawning grounds for Cisco and Lake Whitefish. Now, of course, recovery of these native prey fish would benefit the Great Lakes food webs really in a good way. But survival bottlenecks, as you have heard, are occurring at early life stages. Who knows, they may have always been at that stage, but we're going to look a little closer today at Chameau Bay larvae, and I'm going to cover three particular topics: segregation of nursery habitats, abundance, variability, and asynchrony, and weather conditions affecting larval abundance. Next slide, please. So what have we done? Well, for the last 20 years, we have sampled gorgonid larval abundances from a random grid in Chameau Bay and along the shore in the bay. We used ichthyoplankton tows offshore and larval seines onshore, and these have been standardized to catch per unit of effort. We're also using geostatistical interpolation within the bay to give us the best estimate of bay-wide populations for each sample year and species. As you can see in this example map on the right of Lake Whitefish in 2019. Next, please. So what have we found? Okay, so first, Cisco and Lake Whitefish are virtually identical. We know this. And we used the genetic analysis, and when I say we, I mean Pixar and Wendy Stott, to identify the larvae to species. This was first done with a relatively small subset of a few hundred fish from the 20-year archive by Wendy Stott. That's been published in 2020. And then in this last year, Nick and his crew processed over 2,500 larvae from the full-time series. The bar chart you see on the right, you read the values from the left vertical axis. The top one is for the offshore sampling of larvae, and then the bottom one is for the onshore seine sampling. And they show the number of larvae identified as either Cisco, excuse me, in the orange, or Lake Whitefish in the blue. And the results clearly indicate that there's segregation of these species nursery areas, with essentially Cisco occurring offshore and Lake Whitefish occurring onshore. There's very little overlap, and the dominance is consistent from year to year, with just a few minor exceptions for some of the onshore sites. Now, the black dashed lines indicate the sample larval abundances in each year, and those values are read on the right axis. Note the differences in scale and the variability of those abundances, as well as the asynchrony of the peaks between those two. Now, because of the strong dominance of Cisco offshore and the Lake Whitefish onshore, I'm going to speak about the species instead of the habitat for the rest of this talk. But know that in this case, what I'm presenting here has a little bit of mixing going on. Next, please. All right, something else we also know is that, as Kay mentioned right off the bat, that the sizes of the cohorts of these species are notoriously variable. And they appear to be associated with winter weather conditions. Now, this figure here it's got a lot of information, and it's really just to give you a sense of the variability in winter ice and thermal conditions, which you see on the left. And also to show you the weak direct relationship between larval abundance and temperature or ice conditions in Chameau Bay on the right. Now, the formation and melting of ice are really quite complex, and I'm happy to go back over this later if folks have other questions. But that feeds into this relationship between larval abundance and winter conditions, which are also very complex. It's not a one-to-one relationship. Next. So because of this complex relationship, we applied neural networks to model the connection and to make estimates from combinations of several of these winter condition variables, as well as to provide some insight into the most important conditions for each species. And so what you can see here in the top charts, we have Cisco on the left, Lake Whitefish on the right, and clearly the models fit the highly variable abundance time series very well. The blue lines in each case show the observed abundances, and the orange line indicates the neural network predictions. And note again the asynchrony in the peaks and the differences in the abundance scales. The bottom charts show us the weightings on each of the independent variables that was used to train and test these neural networks. And again, these are the weightings, so they're not variable values per se. These are actually, but think of them as the multipliers used in the model. But they give us some idea of what seems to be the most influential. And these graphs here show several metrics. But just look at the solid line for total weight. Okay, so for Cisco, which again, we're thinking mainly offshore, that's on the left. Winter conditions seem to emphasize low thermal conditions, thermal units, so they like it cold. A high rate of ice formation to 10% of coverage, relatively warm temperatures in February, and then relatively cold temperatures in January and April. Now, by comparison, Lake Whitefish, which again, we're thinking onshore mostly, on the right here. The variables, by the way, are in the same order from left to right. Their conditions emphasize cold December and January temperatures, relatively warm March temperatures, and relatively low maximum ice cover for the lake. Now, I'm sure this will leave you all with many questions, but I'm going to finish up with one more slide, and then I'm happy to address any questions about the methods or whatever you have when we get to the panel discussion. So, Bo, if you go to the next one, please. This slide is not from Lake Ontario, but it is the most vivid picture of the power of ice and water that I have, and that clearly those things are important for Corriganine larvae. In a quick summary from this project, we have discovered several very interesting things. I hope you will agree. Clearly, genetic analysis is required for reliable species identification. Also, Lake Whitefish and Cisco have different nursery requirements, and they may have fewer options than they used to. We don't know. Also, they respond differently to winter weather conditions. The next step in this process for us is to estimate the baywide abundances for each of the species for each year. And in fact, the geostatistical part of that we finished yesterday. So moving forward, there are a couple more things that we need to look at least a couple more things, of course. And one of those is the habitat conditions of the spawning in the nursery areas. And the other is, we need evaluations of options for habitat protections and improvements. And a lot of that work is actually already going on at T-Lobs and by other researchers and partners across the region. So I think we're on our way, and I hope this clarifies some of the things that we have been wondering about for a while. With that, I'll say thank you for your attention, and I'll pass it back over to Bo.
Thanks very much, Jim. And to all the speakers, you all did a really, really excellent job of distilling the key information down within 10 minutes. Everybody was great with time. All the talks are really complementary, sort of a lot of sort of very similar themes. And if we can have all of our speakers turn their cameras on. I figure out how to close this and show. Does anybody want to start us? So for questions, we welcome definitely questions from our large audience. We had nearly 100 people joining today, which is awesome. So for audience members, you can raise your hand, throw your camera on ideally. You can put a question in the chat or just ask it yourself. And we've got Taylor to kick us off.
Hi, can you hear me okay?
Great.
Excellent. Yeah, this has been an excellent time. Lots of questions and topics I love to think about and hear about. So yeah, thank you everyone. I have a question for Steve, which it's awesome to see those data sets. And it's really, I thought it was so cool how your different gears, the Newston versus the Sains, aligned so nicely with the transition from pre versus post yolk sac. And I was wondering if you had done any sort of analysis to try and look at like what the survival rate is in between what you see in the Newston versus what you pick up in the Sain, and how much that varies year to year, with the idea that maybe that's a bottleneck, or maybe a bottleneck in some years more than others. Thanks.
Yeah, no, I haven't, like, I've looked at the catch data very little, and so I haven't really explored that much. And looking at it for this talk, some of it was like, oh wow, that's really interesting. We should have been looking at this for beforehand. So I haven't really explored much of that. So that's out there, and definitely is something that's worthwhile looking at. I hadn't realized until I was looking at this that we really aren't sampling those fish very well until they've lost their yolk sac. And so that was really surprising to me, because we do catch some, but just not really in proportion to how many there must be. So I haven't explored that, but that's definitely a good idea, Taylor.
And as a follow-up, Steve, like, have you talked to other people that are doing this Newston Beach seining to see if they are catching them and you're not? You sort of intimated that you had.
Yeah, I'm not sure about in terms of this whole size distribution. I know fish that we've looked at from Lake Huron, there's a lot of yolk sac fry in those fish. Those are more from an offshore than a beach. So I know you can catch them in those nets, but it would be worthwhile looking for all these beach sampling if those yolk sac fry aren't really on the beach and they're maybe further offshore.
Yeah. Okay, thanks. Any other questions for Steve while we got him from the panel members? If not, we'll move on to Alex. Okay, Alex.
Hi, everybody. Thanks for all those great presentations. So my question is for Nikki, which I could just walk across the hall here and ask her, but I think it's better for this. So was I right in seeing in your first figure there with bloater, Cisco, and Lake Whitefish that bloater had the highest tolerance for UV radiation?
Yeah.
What do you think is going on there, right? Because that's like exactly opposite side of the thought.
Yep. That is also what Bo and I think say every single time we that figure comes back up. It's like I don't know, but I don't know. It's not. I think one thing is that we did find in CSMI in 2021 was that we caught a lot of bloater at the surface offshore of Lake Michigan, and that is like the most transparent habitats as far as UV goes, especially. So I don't know if that's just like they've just evolved that way because they hatch, they're spawning and hatching deep, but when they're swimming up, they're still at a stage vulnerable, and so they've evolved that way or what. But yeah, that wasn't what we were expecting either, by any means. So I unfortunately I don't have a lot more about that other than they were there regardless of what the UV exposure was in the lake. They were caught. So low abundances, but they were caught.
Awesome, thanks. Yeah, your explanation of them going up into the kind of the central basin of Lake Michigan, there's lots of UV, that makes a lot of sense. Thank you.
All right, thanks, Alex, for the question. Let's go then to Roger.
Hi, everyone. Thanks for some really interesting talks. I don't know who to direct this to. Maybe it's Nikki, maybe it's others, but I'm just wondering about the proximity of spawning reefs to the nursery areas. And obviously that wouldn't necessarily change over time, but I'm wondering, there's an issue with lower numbers of fry coming to these areas, or given the UV issue, if some other areas where larvae may be swept to are now more useful habitats from a nursery standpoint. Thanks.
I have a quick
Cory.
thought.
Oh yeah, I was just gonna say I think you could answer this probably best with your data.
Oh, it's a really good question. It gets me super fired up. So some of the site selection for beaches, we used historical spawning areas to inform where to sample. Some of it just came down to the beach being easy to access. I'm really curious about the same question for Harbor Beach, because that historically there certainly were whitefish spawning outside of Harbor Beach. I don't hear about it as much on in contemporary times in southern Lake Huron. So, and this is kind of like a lot of what we sample at Harbor Beach is actually in that harbor too. So I've been talking with Mark Rowe and others over at NOAA, looking at their biophysical models and how things move from Saginaw Bay. They kind of tip down the thumb like water down a glass in terms of nutrient flow and stuff out of Saginaw Bay right down through to Harbor to Port Huron. But I'd really be interested to see, or hear about ways that we could look at this closer. What is the proximity to these spawning reefs? Are there still some there? Can we use larvae as a proxy to actually find new ones that maybe came back online? I don't have an answer, but I'm certainly excited about the question.
Yeah, I wonder, yeah, go ahead, Jim, because your data are really relevant too, given
Well,
Chameau Bay and the spatial segregation. Yeah.
Right. I was just going to follow up with a little bit. I mean, I don't have an answer that's any better, really. I could say Chameau Bay is a relatively small water body, and we know that there are 3 reefs out in the middle, and only a kilometer or less from most of the shoreline. The question has to do with, are the larvae being transported from those places to the shore? Or are they coming out on the shore and being moved around in other ways? The lake or the pond there, it's pretty much a big pond, is really affected by waves. And I believe there's a hydrodynamic model that's being developed. I think Brian Weidel can tell us more about that. For Chameau, to tell us a little more about the transport potential. But the other thing I think Jon mentioned that I think is fascinating is the vertical diel migrations, right? Because if you have different current regimes at the surface during the day that you have at night, you can either be moving far or you can be shuffled back and forth. So yeah, so Roger, I don't have a much better answer for you than that. The other thing I'll say is we're seeing some fascinating things by looking at the geostatistics, which gives us an interpolated map. And there are some areas which look like they may be really good for producing like whitefish that are probably very highly developed human-wise. You know, there's a lot of docks and things like that, and places that we couldn't actually get in with the seine, but they could be very good habitat for all of them.
Yeah, thanks, Jim. Kay, I wonder, or Steve, just because I see you, like, so Cory mentioned that the sites he chose were like nearby historic spawning locations. And I wonder, how is that frequent throughout sort of the monitoring, to any of your knowledge, like where you are doing your, Steve, or is it more just sort of, this is a good beach, it's good access, or same thing for Kay and some of the work they're doing in their 4 sites?
Yeah,
Yeah,
our our sites were chosen because they were access. The Muskegon one, we knew there was a fishery there, and we're there, so it was sort of a combination. We knew there was spawning there, and so it was chosen because of that. The other ones were chosen because of easy access, but we also knew there was spawning going on in the river mouths, for all of our sites.
Yeah, thanks, Kay.
yeah, ours were pretty similar. All 4 of our sites, so they're all easy access, but that wasn't the only consideration. That was mostly how we chose the exact spot within like that general area. And for everything except Duncan Bay, over by Sheboygan, we knew for sure that there either were fish currently spawning in those areas, like very nearby, or like historically there had been. So Little Traverse Bay, Sturgeon Bay, and Big Stone Bay, we knew, either currently or had in the past. And then Duncan Bay, currently we didn't know, but in the past, and so that one got added.
Yeah, so good question, Roger. I mean, the really curious thing to me is that certainly these whitefish do seem to be finding their way deliberately, or I would think it's deliberately, like towards these very shallow beachy areas, whereas we're not seeing nearly as many Cisco there or any other species. And then in terms of like day versus night, which was mentioned, maybe quickly before we go to Frank, I wonder, Jon, do you all, like, do you and Simon think that's a catchability thing where like half of the fish are able to avoid the net during the day? Or do you really think that they are like below the net, on half of the population is below the net um, during the day, and then more of them are up in the water column at night? If you had to sort of make a hypothesis.
Yeah, it's a really good question and something that we've spent a lot of time, well, myself, Simon, and others spent a lot of time just thinking about. And I'm sure it's probably a combination of those things. And also, as mentioned also by Jim as well, it's just, thinking about like the wind and currents, on nights where there's not as much wind, maybe they're able to actually move more as well. So if they want to get to shore, they might have more of a chance. But yeah, I imagine it could be, UV radiation, as Nikki mentioned, maybe that plays some role, trying to avoid that in really shallow waters. Following food. So we're finding with this work too that the zooplankton densities are higher on the beach than they are in more nearshore waters. Finding food, maybe trying to avoid predators. But just like observationally, sometimes with a noosa net, if you're capturing larval fish, not always, but sometimes you see a bunch just right at the bottom. So, thinking even with the noosa net, are we missing some during the day because they're just right at the bottom, or they can kind of scurry out quickly or get underneath if they're close to the edge, where at nighttime they might not have that opportunity. So I imagine it's a mixture of those things, but definitely interesting to explore more for sure.
Yeah, just Jon, you said something that got me thinking too of there's a decent amount of like inland lake literature, at least from my advisor from Craig and his team, where looking at diel vertical migrations with zooplankton, different species have different tolerances. Copepods generally seem to be that they have more resistance to UV than the calanoids or Daphnia. So there's some dynamics there that I've been thinking a lot about with these presentations too. And if the fish are following the food and the food's avoiding UV, is that in some way, how does that play with their own like tendency to, try to go to the light or not? And that's, we didn't add that in our experiments, but I would be really curious if anyone did, to see what they found.
Yeah, good points. Really great discussion. All right, let's see where Frank wants to take us. Frank.
It's remarkable with all this new technology, scientific breakthroughs, the amount of information out there is just unbelievable. And I'm asking this to anybody that would like to answer it. How about AI? Is there an effort through the scientific community to start incorporating AI to better summarize and analyze the data? Thank you.
Okay, that's not where I thought we would go, but that's a good question. I don't know for our panelists or anybody else
Yeah,
that's
well,
that's viewing, please weigh in. Yeah, go,
I'll
Jim.
take a stab at that. Okay, so Frank, AI is a pretty general term that's been thrown around an awful lot these days to describe many different things. But I will say the neural networks I showed are a type of AI, and they represent pattern recognition, right? That's what a lot of AI does, is pattern recognition. And so you may consider that some sort of start. There's no concerted effort that I know of to incorporate that or other aspects of AI in data analysis yet.
Thank you very much. And you would think that that would be something that would start really coming together because there's just so much information out there. Thanks, everybody. This has been great.
Thank you, Frank. Anybody else want to weigh in on that topic? I know there are a few people. I was looking to see who else is on here that I might just call out, but maybe I won't do that. Anybody else want to answer that question that's on the call today?
I put it in the chat, but I know Nick Sard at SUNY Oswego, he's like amazing with his incorporating AI and stuff into some of his just routine approaches, as simply as just recording transcripts of meetings and being able to share back notes with these large collaborative efforts. I think that has been really impressive to me. Like Kai mentioned at the start, right? It's hard to get so many different groups together and coordinated and all on the same page. And just having a tool like that that's kind of writing the script for you or writing up the conversation for you as you're having your meeting, instead of having to go back through and scramble or to put your notes together and make sense of that, and sharing that back with everyone else of the team that they can refer to it. It's really helped, I think, enhance some of the conversations that we have with him and his researchers, his team. So that's one way or example, which I really have seen. The use of AI, I think, help with a lot of these huge coordinated efforts.
Thank you.
Okay. So I'll just maybe hit on a few notes I took down. So Jon, I think your experiments are really cool. At your 1 per liter as your low, which is pretty typical, I think, for what's been observed in many of the beach densities I've seen, which are still higher than what's seen just a little bit offshore, as was mentioned. And then sort of Cory stuff showing this differences in growth rate between sites with high and low zooplankton, which is sort of consistent with that. But even your highest survival, and you sort of made note of this, was only about 25%. And I guess you're doing it again this year, but were you surprised, like, of the essentially ad libitum food, your survival was still only 25%, or is that, am I being too critical of that?
Yeah, I would say we were surprised. We expected just some natural death, I guess, to, mortality to occur just from stress or, whatever, being in the tanks, different environment. But yeah, we were, I guess, we didn't expect how many would survive, but I guess I would expect it maybe more, at least in the high treatment. But 20 per liter, that was also part of that. Well, most of that was from sort of like the gradients we found just from the samples. 20 is still sort of low for or still pretty high for what we observed. kind of on the higher end. But there's also like a previous study that found that about 20 zoops per liter was a needed amount for a different cragginine species, I think in Europe to even survive at least a little bit. So I don't know, maybe, it's interesting. We'll repeat it again and see if it's similar or not. But, is 20 sort of like a threshold or is it lower or higher? It's sort of a question with that for sure.
So you'll use the same densities this year?
We're using the same low and the same high, but the medium, we're actually increasing that a little bit
Okay.
to see if there's some sort of like step between, it's 10 and 20 basically. Because right now, the first time we found low and medium to be, very low, like both of them, and high being higher. But is it more of like a stepping sort of a level to kind of separate to make the medium more in between the low and the high versus more towards the low? We thought about changing the high. The hardest thing is we only get so many zooplankton. We're
Mhm.
collecting them from the river, and so we need to get enough volume of water. And sometimes getting 20 per liter to fit all of the tanks is difficult. So we also are kind of constrained logistically with what the highest could be, basically.
And I love that your study also has Lake Superior in it, where, of course, so we have North Channel without mussels or low mussels and Superior. And if the mussels are affecting the food, Cory's study showed higher densities of zooplankton, of course, which you were a big part of, in the North Channel relative to southern Lake Huron. And what about the Lake Superior sort of beach densities of zooplankton? Are they also like lowish or any different from, say, northern Lake Michigan where it's quagga infested?
Yeah, I guess we haven't looked at that specifically yet by the zooplankton densities by lake, but definitely something we'll be doing and would be interesting. I'm not, I think from Simon had a previous manuscript from his thesis, but I think from that they're pretty similar. Um, like perhaps Michigan and Huron were higher, but now they're getting lower to where Superior kind of is type of thing. But for this study, we didn't look at that yet specifically.
Interesting. Okay. Other questions you all want to ask of each other? Not, it has been an hour and a half. This has been a really great set of talks and good discussion. Nikki, you gonna ask something or you just no?
Oh, I was gonna ask Kay what their thought was for some of those long-term declines. If there's any one driver that your guys' team is really like queuing in on right now, more so than not, and or just curious, like the historical knowledge from the team. Is there anything from the community members or anything like that that have kind of like really pinpointed or really is driving some hypotheses right now?
Good question.
Yeah, so the thing that I think of immediately for declines, I'm thinking of Little Traverse Bay. So that's where we've been doing most of our like egg take happening, and we do our beach sampling, walking distance from where we do our egg take, right? It's just down the beach. And we've been finding lower and lower, numbers of whitefish when we're trying to do that egg take. We're also seeing not very many babies showing up on the beaches. So those things are logically very connected. And it could be an artifact of that. Obviously, that doesn't affect our Little Traverse population, wouldn't hold the same for our other places, and we do see those declines at our other sites as well. But maybe that means that they're all declining. Uh, but I could really only say I am very sure that Little Traverse Bay, the adults are declining, and also the larval fish on the beaches are declining. The others, we haven't done those types of studies there, but that would be something really interesting to note. Um, I'm really interested to see what we see this year in terms of fish in these areas because of the ice cover. Last year, Little Traverse Bay never froze over. This year it has. And so there's some thoughts of ice cover, even for, UV stuff like you're doing, or, protecting them like that, like ice cover being really important there. So I'm really interested to see if we can do it, last year to this year comparison, just because it was so different.
Yeah, I'm excited for you guys too, getting that ice. It's a nice year.
Yeah. I think everybody enjoyed, humans too, enjoyed having a winter. And Kay, I don't know if you have your chat open, but Becky Adams provided another link where maybe Cory could add to the map about some on the Saugeen Peninsula, the Saugeen Ojibwe sampling along the shoreline. So thank you for that comment, Becky. All right. Well, thanks again, everybody. Reminder, if you would like to volunteer to give a talk in April, please reach out to me. If we don't connect in April, we do have a great set of talks by Caitlin King and Laura Lee talking about some restoration planning for the Cisco restoration that is going to be beginning in Lake Erie, in the New York waters of Lake Erie. So they'll be talking about that in early May. And with that, we will close off. Thanks to Ted Tresca for pitching in and providing the behind the scenes support from the Fish Commission. And thanks again, everybody that's still on, give a round of applause or congratulations to our excellent speakers. Have a great weekend.
Thanks, Bob.